Morning, everyone. Ooh, there we go. I would like to welcome you to San Antonio. Thank you very much for joining us for one of the first sessions of the conference here at the Basic Science Workshop. My name is Alana Whelm. I'm Senior Director of Basic Science and Professor of Oncological Sciences at University of Utah, Huntsman Cancer Institute. We have both a live audience, as you can see, and a virtual audience today. And so we'll be taking questions from the online app if you wish to enter them that way, or they will be bringing microphones to the room. We're just waiting for those, but we're going to go ahead and get started. So our first presenter is Dr. Reuben Harris from the University of Texas, San Antonio. He traveled really far to get to this meeting today. The title of his talk is APOBEC and Genome Evolution in Metastatic Breast Cancer. I want to give a little background on Dr. Harris. He's an HHMI investigator focused on understanding fundamental mechanisms of tumor mutation and evolution. He received his PhD from University of Alberta and did his postdoc at Yale University and also at the Laboratory of Molecular Biology in the UK with Dr. Michael Neuberger before he started his own lab, first at the University of Minnesota. He became an HHMI investigator in 2015, and then he moved to University of Texas, San Antonio here in 2022. He's currently professor and chair of the Biochemistry and Structural Biology Department. His work is really fantastic. I'm looking forward to this talk. His lab is really seeking to understand how DNA mutating enzymes, APOBECs, provide immunity against viral infections, yet also contribute to tumor evolution through DNA mutagenesis. So we will go ahead and let Dr. Harris give his lecture.
All right, thank you very much. First, I want to thank you all for getting up so early this morning and helping us open this session. That's a real treat. There's a lot of extra seats over on this side of the room if you want to come on over. So I view myself this morning as an opening act, okay? There's 3 great talks to follow, and I want to keep it light. You guys are still absorbing caffeine. An alternative title to what I want to talk about today is Goldilocks and the 7 APOBECs. There's no real fairy tale that explains this story perfectly, but it is a little bit about a particular trying to find just the right level in this particular talk, and there's 7 of them instead of 3. My disclosures are here, including the most important one, as Alana said. I've moved to the University of Texas Health Science Center in San Antonio, literally 20-minute drive from here. And happy to be now a local. But I feel like I've grown up with this meeting in the last 10 or so years as well. So because this is a little bit of an introductory session, and this is kind of a landmark year for APOBEC enzymes, I wanted to give you all a little bit of brief history. So 20 years ago this year, we actually discovered the DNA mutating activity of these enzymes while I was a postdoc with Michael Neuberger, and this is Sven Pedersen-Martin and I working together. First on AID, showing it was a DNA deaminase important for adaptive immunity by antibody diversification, and then second, with this molecular cell paper, one can see with the various different APOBECs. And we suggested that these results reveal the existence of a family with possible implications for cancer. And it took another 10 years, and in my lab, a few brave graduate students, along with a cohort of supporting scientists to get a handle on these enzymes in cancer mutagenesis. And so a paper from us and then some sequencing papers as well described a mutagenic process attributed to these APOBEC3 enzymes. And when we first started coming to this meeting, actually about 10 years ago, I didn't look back in the history books, but it's close to that, this was a foreign word, and very few here knew all about it. But I hope anyone who sequences tumors, especially breast cancers, now knows about it. So this is using APOBEC3B as an example. It's overexpressed. Breast cancer here is in the middle, so in about half of all breast cancers. Some tumor types have APOBEC3B overexpressed to even higher levels. Its signature is evident in tumor genomes. So breast cancer is shown here, and the APOBEC signature we define as and this is really conservative definition because it doesn't account for all non-signature outcomes, but the signature is C to T and C to G mutations in TCA and TCT motifs, and you can see that here with a pileup of mutations in TCA and a pileup at TCT, and there's both C to T as well as C to G in black. And it's clinically relevant. So in collaboration with John Martin's lab, we've published a few papers, and others have published in papers, but essentially high levels of APOBEC3B in primary tumors predict poor clinical outcomes. And this is with estrogen receptor positive metastatic breast cancer. And so levels in the primary tumor predict poor outcomes following recurrence. And in this case, because it was a relatively older cohort, but still with excellent data, it's in the presence of tamoxifen as a monotherapy. So APOBEC impacts many cancer types in breast cancer. This is still one of my favorite slides. This is borrowed from Professor Martin's, and it's an analysis of a Sanger Center data set of primary breast cancer and metastases in a Dutch cohort. And I just want to point out a few features here, but if you look at the most common mutational processes in breast cancer, there's aging, which is spontaneous deamination of cytosine to uracil, BRCA, which is defects in homologous recombination repair, BRCA1, BRCA2, and APOBEC shown in pink with the two signatures depicted in different shades of pink. And I think what you can appreciate here is that this, the mutagenic component or contribution changes from primary breast cancer to metastatic breast cancer, with the BRCA component increasing, which is consistent with its role in tumor evolution, and the APOBEC component massively increasing from primary to metastatic breast cancer, again arguing for a role in ongoing tumor evolution. So our working model for APOBEC mutagenesis in cancer is here, and we've been testing this in various ways over the years, but essentially these enzymes are expressed in low levels in normal mammary epithelial cells, and in normal epithelial cells of other cancer types, and then through inflammation or cell cycle dysregulation, or in some tumor types, not breast cancer, but virus infection, APOBEC levels can go up, and that results in increased mutagenesis and increased opportunities for tumor evolution. So the APOBEC family, and why I've been using it in the singular thus far, is a little bit complicated. So there's AID, the antibody gene deaminase that I alluded to initially, APOBEC1, the namesake, and then 7 APOBEC3s in humans. And so one of the big questions we and others have had over the years is which ones are contributing to this massive mutational impact in cancer. And just to orient you also, this is a superposition of APOBEC3A and APOBEC3B with single-stranded DNA in the active site, and you can see the target cytosine there. And the -1 nucleobase is the reason these enzymes have a TC preference, and that's what we see all over genomes, and that's part of the APOBEC, the conservative definition of the APOBEC signature. But 6 out of 7 of these, are actually 7 out of 8, mutate in TC motifs, and so it was a real important question to figure out which APOBECs are actually responsible for that. So I'm going to address that in the first part very shortly, and then go on to some novel models that we've tried to develop to address it preclinically. So these are 2 recent studies. I want to highlight this one in particular from Mia Petchak and John Masajowski with a supporting cast, where they've done APOBEC deletion studies in human cell lines, including breast cancer cell lines. And our study that's still lingering on bioRxiv, hopefully soon out, that shows that APOBEC3B, the impact of APOBEC3B and A in a human cell line. And both of them arrive at the same conclusion, which is APOBEC3B and A are capable of inflicting that signature I just described. So in this system, we established what we call a mother clone, which is a clean cell line that expresses the herpes simplex virus TK1 gene as a selectable marker. And then we can introduce the various APOBECs, and by subcloning and drug selection, we can assess the frequency of mutation. And here's the expression of APOBEC3A, B, and H with controls. And it's very apparent just by simple selection and colony counting that APOBEC3A and APOBEC3 increase the thymidine kinase mutation frequency. And an additional value of this model system is one can then sequence that mutational reporter, and one can see that in comparison to the controls, where there's lots of black and blue ticks, in the APOBEC3A and APOBEC3B expressing conditions, there's lots of red ticks, and those are APOBEC signature mutations in the canonical motif. So that said that they are capable of damaging the genome and inflicting its signature, but then one can take those clones and sequence genome-wide, and now one sees what is the textbook signature of the APOBEC enzymes, and this is also called cosmic SBS, or signature 2. and this is, that's the red one, and cosmic signature 13, the black one. And you can see these C to T and C to G mutations in APOBEC3A and APOBEC3B expressing lines, genome-wide. And when one takes all of those mutations in the canonical TC motif, you can see that these motifs are very, very similar. So TC, ART, TC, ART. But if one looks at the -2 position, there's a bias toward pyrimidine for APOBEC3A that's not apparent for APOBEC3B. And when one uses this broader signature to infer which APOBECs are responsible for mutagenesis in breast cancer, actually both is the answer. So there's a cluster of breast cancers that have signatures that more closely resemble APOBEC3B in blue, and a cluster that resembles those that, breast cancers that have signatures most similar to APOBEC3A in red. And in reality, APOBEC3A has that 70% pyrimidine preference. Very few of these have that high a level of pyrimidine at -2, suggesting that both enzymes are actually contributing in most of these breast cancers. And there's a whole load of breast cancers not shown on the page here that have higher levels of course, SBS3, which is an indicator of homologous recombination repair deficiency, not APOBEC. Okay, so we've got about 10 minutes remaining in this, and I want to shift gears to how are we addressing this in mouse models. So it's very important to note that humans, most of us in this room, I can elaborate in questions if you like, have 9 APOBEC family members. A few of us may have a couple of SNPs that compromise our APOBEC locus a little bit, but mice really have an APOBEC deficiency. They only have one APOBEC3, and it's an orange-blue double-domain deaminase. And I neglected to say that humans have 4 double-domain deaminases, B, D, F, and G, and then single-domain enzymes A, C, and H. So it's very different. And not surprisingly, for any of you that have sequenced mouse tumors, they're not as exciting as human tumors usually, right? They're pretty, what I call vanilla, right? They're cleaner genomes, much, much fewer mutations. So we would really like to be able to address several questions, as well as develop preclinical models for APOBEC mutagenesis and further studies. So they don't have APOBEC3A and B, so how do you get them there? There's different ways. The first way that we used is a hydrodynamic tail injection model, where one can inject DNA plasmids. Those will be taken up by liver cells spontaneously. You can accelerate this process if you put your DNA cargo on a Sleeping Beauty cassette, a transposon cassette, so that it hops into the genome at a higher frequency. So we can enroll these animals at birth and inject them with different APOBEC constructs after 2 months, and then withdraw an inhibitor called nitisinone from the drinking water, which causes an accumulation of toxic tyrosine degradation products and would otherwise kill the animals if they don't take up this DNA cassette. So using this elegant system that was reported first quite a few years ago by the Grompe lab, we're able to introduce the various APOBECs. And this is an immunohistochemistry of APOBEC3A in liver cells, so we get between 5 to 10% uptake. A lot of those cells are also showing higher levels of the DNA damage marker, gamma H2AX. And that's 48 hours post-injection. But if one then waits 6 months and then looks at those livers for evidence of tumorigenesis, there's actually a high level of tumorigenesis only in animals that have been injected with APOBEC3A constructs and not with any of the other human APOBEC3 family members. So this is a very interesting observation, number one. What was additionally interesting, and we learned very quickly, is that unlike those cells 48 hours post-injection, the tumors are now negative for APOBEC3A staining. Some of the surrounding normal hepatocytes are positive for APOBEC3A. And this suggests that it was there initially and then disappeared, but we actually know it was there in part of the tumorigenic process, because when we sequence those tumors, there again is the strong APOBEC mutational signature with a strong bias toward TC and a pyrimidine bias at the -2 position. So APOBEC3A was there, it was active, it damaged the genome, and probably selected against itself. We also know that this selection is not due to anything in our genetic construct, as a single catalytic site mutation is now tolerated as the liver repopulates. So this is a catalytic mutant E72A that's not selected against and not at all tumorigenic. So I didn't put those data here, but it's essentially in the spontaneous baseline. So we know that A3A is sufficient, catalytic activity is required, the APOBEC mutation signature is very abundant in those tumors, And in these conditions with 6 months, other APOBECs are non-carcinogenic. But importantly, if you wait just a little bit longer, and this is consistent with the biochemical activity of APOBEC 3b, which is a little bit kinetically about 5-fold less efficient than APOBEC 3a, it also becomes penetrant, and the others don't. So B takes longer, and we're working now to characterize these APOBEC 3b tumors in this model. And of course, it's a whole reset and another year and all that sort of fun stuff to collect enough for significant analysis. But I think both are mutagenic in this particular system. So that's what I just said. They catalyze signature mutations. Catalytic activity for APOBEC 3a is required, and this is consistent with what I would call mutational Big Bang, where we put a lot of DNA damage, a lot of selective pressure on the system. Obviously, it inflicted tumorigenesis, so it tests our central hypothesis that these are tumor drivers and not simply passenger processes. And we're really excited about characterizing this further because it is an isogenic system, and we can analyze mutants and mechanism. So, but I wanted to bring you back to the Goldilocks and the 7 APOBEC story, because there really is a just right level, and that sort of bodes to this assumption that we have, where APOBEC 3a is not tolerated for a long time in those hepatocytes. So another way to introduce APOBEC to mice is by a classical genetically engineered mouse model. And so here we've knocked in the APOBEC3, human APOBEC3b gene into the ROSA26 locus, and we've included a strong triple transcription stop cassette so that it is off unless induced by Cre recombinase in various tissues. Unfortunately, so we can get expression in various tissues using this model. We can get it whole body, we can get it inducibly. And as described in these papers, additional tweaks are required to elicit phenotypes, but APOBEC 3b alone doesn't have a Kaplan that's different than normal wild-type mice, C57 black 6 mice. So this low level of APOBEC 3b isn't sufficient to elicit what I was just describing, at least in a normal mouse lifetime and without additional modifications and selections. So that inspired us to go to bring our game, up the game a little bit further, and now use that same triple stop cassette with APOBEC 3b, but knock upstream of that a CAG promoter, which is a strong heterologous promoter that won't express APOBEC 3b until Cre induces it, but after at higher levels. And you can see that down below. In comparison to our ROSA model, there's lower levels of APOBEC 3b and now higher levels at the protein level here, and also higher levels at the activity level. This is a DNA cytosine deaminase activity assay with single-stranded DNA. We can see robust staining in the nuclear compartment of many different cell types. Some of those are shown here, including lung cells, intestinal cells, spleen cells, breast epithelial cells, I should have zoomed in on, but also there, of course. And in contrast to the ROSA model, which is a different data set plotted here in comparison to the control, higher levels of APOBEC 3b are penetrant. And to put this in perspective, so we're really excited about this result because a normal mouse lifespan is here, normal human lifespan 75 or 80. This is equivalent to 50-year-olds getting cancer. And the types of tumors that penetrate in a full body model are mostly blood tumor types, liver tumor types, some lung, and a smattering of other tumor types. If we drive it, we're working on driving it specifically in epithelial cells, but that's actually easier said than done. And some of the classical models out there, like MMTV Cre, actually result in leaky expression in the blood compartment, and we have penetrance there too. So we're working on driving it also in the mammary epithelium. But what's really additionally fantastic about this model is that the tumors are really heterogeneous. So unlike many different mouse models where there's homogeneity if you're driving a KRAS lung tumor or if you're driving a particular P53 APC-dependent process, they're pretty darn similar. But here there's heterogeneity. So these are different liver masses on the top. So we're looking at different phenotypically heterogeneous tumors. These are various lymphomas down here with massive splenomegalies, a huge large lymph node here, and an example down here. And when you compare this to a primary liver, and so there's a very nice cobblestone appearance of the liver cell nuclei, Some of these liver tumors are really dysmorphic. There's clear signs of atrophy within the tumor masses. One can see blood vessels nearby. APOBEC3B, in contrast to APOBEC3A in the liver model, is strongly expressed still in the nuclei of these cells. And we actually even have evidence for metastasis. So here's a normal lung tissue with some APOBEC3B expression, and here's a lung tissue with evidence of a liver metastasis. So these cells look a lot like liver cells and not like lung cells. And actually sequencing, RNA sequencing and whole genome sequencing confirms that. So we have evidence of liver to lung metastasis and also evidence for multifocal blood cancer development as well. So looking at the mutational levels, so these are whole genome sequences of APOBEC3B expressing tumors. Here's a primary tumor and 2 different METs. You can see the strong APOBEC mutational signature emerging. Interestingly, it's predominantly signature 2, not so much signature 13, which would be C to G mutations in those same motifs. But in terms of how do we know these are true metastases, not independent tumors? So there's shared mutations. So for instance, here's a mutation that's private to the primary tumor. Here's a couple that are common to, that are present in these different lung metastases. And one can play that game and even find mutations like this one and this one that are private to each of the lung tumor metastases, implying that there's ongoing evolution of these cells. So here are some additional examples where we're looking at a normal intestinal tissue and a Peyer's patch. And Peyer's patches, of course, are little immune organs that are important for adaptive immunity. But when you're looking at some of these lymphomas, there's very, very strong, large masses, both in the Peyer's tissue as well as in other lymph tissues. You can see very aggressive tumor growths, again, all positive for APOBEC3B, and this is staining for gamma H2X, indicating ongoing evidence for DNA damage. And again, sequencing these guys, so now there's a lot more mutations, but again, very strong APOBEC mutational signature and shared mutations between these different anatomical sites. So overall, what I hope I've shown you today is that both APOBEC3A and B are potent mutators that can induce the APOBEC mutation signature in human cells. The signature, the composite that we see in tumors and in metastases is likely to be a combination of the two. They both drive tumor development, and they're not simply passenger processes. Human APOBEC3B expressing mice have an enormous heterogeneity and evidence for ongoing evolution and metastasis. And we're still trying to find the right word for this. So I quoted a few of our colleagues here. These may be master drivers, Fabrice André, or drivers of drivers, as John Martin suggested. I call them enablers, but they are fuel that promotes a lot of these other events that we're interested in studying. I want to thank my team for contributions to these studies over the years. Developing new mouse models is a long, heavy process, and fantastic collaborators from all over the place in our supporting agencies. Thank you all for listening this morning.
Thank you, Dr. Harris, for a fabulous talk and also for staying perfectly on time. So we do have a couple of minutes for questions. I cannot see if the mics have arrived, but you can enter questions online, and I can see them here. There's also people bringing mics around, so
yes.
raise your hand or stand up.
So there are mics there, and if you're, and maybe start with our co-host over here who, since she's got a mic right in front of her.
Yep, go ahead.
This is a really interesting talk. I'm curious as to whether you looked at tissue from animals younger before you saw tumors and whether you saw increases in senescence in any of these tissues.
That's a great question. We haven't looked at senescence yet, but we have looked at younger animals and for expression and development. One thing I didn't have time to tell you is it's a male sterile phenotype also, but surprisingly tolerated down the female lineage. So I'm sure we're pushing the constraints of the just right level in this model. I'd love to do that. Maybe.
Question in the back, please stand up and introduce yourself as well.
François Aymar from Brackman Therapeutics. My question is about, you saw loss of the APOBEC3A overexpression, in your model. Do you see that, or do people see that also in clinic? What about APOBEC 3b? And my question, the background of the question is, APOBEC 3b overexpression leads to sensitivity to DNA repair inhibitors such as ATR inhibitor. So could we treat patients with an ATR inhibitor, or are they likely to have already lost APOBEC 3b, or are they going to lose it so fast that it's pointless to treat them?
Yeah, so if I understood the question, it's regarding expression. So there's a little bit of both there. So in actual human tumors, at the RNA level, there's much higher levels of APOBEC 3b than A. We've done a lot of immunohistochemical staining, and APOBEC 3b appears consistently expressed in tumors with APOBEC signature, although we need to systematically match that up in a larger cohort, which is in process. We only recently developed monoclonal antibodies to APOBEC 3a that are sufficient for immunohistochemistry. So if you're just looking at RNA-seq data or RT-qPCR data, it's at very low levels in tumors. So you have low levels of a very hot enzyme and higher levels in the nucleus of a not as hot enzyme. And so both are contributing. So there might be evidence for selection against A in tumors if you take absence of expression as selection against, but I don't think so because we never see mutations in APOBEC 3A. But there is, so short answer is it's likely a composite, okay? And there's evidence for inducibility and fluctuations of both APOBEC 3A and B levels with different mechanisms as well. So it's complicated.
Question here?
So very nice talk. I'm Guido Krömer from Paris. So I have a question with respect to the immunogenicity of APOBEC-driven tumors. In mice, when you overexpress the transgene coding for APOBEC3B, do you see some signs of immunosurveillance, and do these tumors develop more quickly in immunodeficient mice?
So this is a great question, and we need to actually have a look at normal tissues to start addressing that question. We're not seeing any overt inflammation and immune responses in different tissues, but we really need to dig deeper now that we have that model. It's possible too, we also need to dig deeper into the actual tumors themselves, which have higher levels of mutation that are now clonal in many cases. So we're interested in pursuing that.
I don't see it. Please stand up if you have a question. And in the meantime, I actually have a question.
Sure.
So the C to T mutational signature, I think, if I remember correctly, has implications on DNA methylation. And I'm wondering if you can speak a little bit to whether you've seen consequential RNA expression changes that might also drive the tumor development, and how you might separate those two things.
Yeah, so with APOBEC enzymes, there's the potential for epigenetic alteration as well. So deamination of methylcytosine to thymine, as well as RNA edits that could contribute. We don't see RNA edits with APOBEC 3b. APOBEC 3a, though, can make RNA edits. And in terms of the methyl, altering the methyl epigenetic marks too, I think both can do it, but A is a little better at it. We haven't looked globally in those tumors yet to see if we have changes that are also to the epigenome yet. That's future also. Yeah, we had to get the model before we could do anything. So this is maybe 2 years from now.
Well, it's beautiful work. So let's thank Dr. Harris again for a great presentation.
Thanks very much.